Today we're going to discuss about intragenic gene interactions. So as we have already seen the intergenic gene interactions. So the intergenic is the interaction takes place between the two genes, that is two alleles present on the locus of two genes, that is present on the locus of the genes, that is the gap between them. But here the interactions takes between the alleles of the same gene, that is the allele at the same locus, they have an interaction. So they are called as intragenic or intralocus gene interaction. So how it can be described means I am giving an example that you are talking to, that is you are talking to yourself. How means you have a mind voice and you have interpret yourself, that is this you have to say to the particular person. Before that you will make an analysis about the topic, so that interaction takes place between you itself. So that is called intragenic interaction. So if a girl has a doubt in a subject, he has to go and ask to the teacher. Before asking the teacher the doubt about the doubt, he or she will be he or she will be estimate how to ask. So what I have to ask to the teacher, the girl will estimating, that is analyzing. So analyzing means interacting to herself or himself, that is known as intragenic gene interactions. That is interactions takes place between the alleles of the same gene, that is the alleles at the same locus is called intragenic or intralocus gene interaction. It includes the following: incomplete dominance, codominance, multiple alleles, and pleiotrophic allele genes. And we're going to see them with the examples. Here they had given only two things, that is incomplete dominance and codominance. So in the that is we're going to see this. The incomplete dominance, the best example is flower color in Miraplus chalaba, and its ratio is 1:2:1. And flower color in snapdragon antirrhinum species 1:2:1. And in codominance, the best example ABO blood groups that we have already discussed in the zoology class. So, and its ratio is 1:2:1. First, we will see about the incomplete dominance. No blending of genes occurs in this. So, we can say it as no blending of gene also. So, this was given by Karl Correns, German botanist, in 1905. What he has done is, he took the 4 o'clock plant, Miraplus jalaba, that is at evening 4 o'clock only it will bloom, the flower will be bloom. So it contains the name as 4 o'clock plant. So what happened? He took them homozygous, red color parent. Homozygous means same color, that is R1 is the gene for red color. And he crossed the red color flower with homozygous white color R2, that is the gene is crossed. But in F1 what happens means, in F1 you get an heterozygous R1. So what color has to become? Whether a white color or a red color has to come. But here you get another color known as pink color, that is an intermediate color came from this cross. Okay, the intermediate color has appeared from the cross. So it is called incomplete dominance. So we will see the definition. When one allele is not completely dominant to another allele. It shows incomplete dominance. Such allelic interaction is known as incomplete dominance. That is, one allele is not completely dominant to another allele. So the R1 contains red pigment and R2 contains white pigment. But in F1 what you are getting? A pink color is coming. So R1, R2 is there, but they are not completely dominant over another allele. Okay, that is known as incomplete dominance. So you think in a class there are two students, two students are toppers in the class throughout the year. So the teachers and the entire management are expecting the two students will get first compulsorily. They are overconfident that they will get the first mark. The both of them will get. But what happened in the board exam? A third, that is who had not expected student had get the first mark. So this is called, this example can be called as an incomplete dominance. So we had expected any one of the students who got throughout the year first, but in the board exam the third person is coming. So that is the ratio, that is the example I had given, and the ratio for incomplete dominance is 1:2:1. 1 red is to 2 pink is to 1 white, and the genotype 1 R1 is to 2 R1 is to 1 R2. So we will see this in detail with the help of the cross. In the first generation, that is in the parents, here the R1 red flower is crossed with R2 white flower. So it is crossed, and the crossed after in F1 generation you are getting the pink color flower. That is pink color, either of, that is the genes is R1 and R2. What happened means, that is as per the Mendelian, the dominant character has to be exposed. Okay, any one of the gene characters should be exposed. So if it exposed means it is the blending inheritance. But here it is not exposed. No blending of genes occurred here. So a new character is appeared. So this R1 is crossed with R2. In F1 you are getting R1, that is the pink color. Now this pink colored, that is R1 is selfed, and in F2 generation you get one red, okay, one red color R1 and one white color R2 and two pink color, that is two pink color R1. Okay, this is the thing appeared in F2 generation, that is 1 red is to 2 red is to, 1 red is to 2 pink is to 1 white. That is 1 R1 is to 2 R1 is to 1 R2. This is the ratio. Okay, 1 is to 2 is to 1. That is 1 red is to 2 pink is to 1 white is appeared. How this takes place? So why these genes are incomplete in dominance? Why means this R1 that is for a particular gene or alleles, two copies are there, that is R1. To the particular character has to expose. So this R1 gene is capable of producing a protein, that is the enzyme, the protein to synthesize the red color. Okay, and same way this R2 cannot able to produce the protein, and they cannot able to synthesize the white color. So what happened in F1? you have one R1 and one R2. So for the protein synthesizing, that is to activate the particular enzyme to synthesize the protein, two R1 genes are needed, that is two copies are needed. But here how many how many gene, that is how many copies there? Only one copy R1. So 50% only you are getting. So another 50% you are getting from the R2, that is white color flower. So because of that, it cannot able to expose the complete character of the it cannot able to express the complete character, that is the red color. So 50% of R1 and 50% of R2 gives rise to the pink color. That's why you are getting the ratio 1:2:1 ratio, that is 1 red is to 2 pink is to 1 white. So this is the interpretation level, that is the explanation, that is international, how the intermediate heterozygous phenotypic characters appeared at the molecular level. So what I had explained, that is the same thing was here. You can see, I will read it and you can understand. So the gene expression is explained in a quantitative way. The wild type allele, that is red R1 has two copies and it produces the functional gene enzyme red pigments. The mutant allele also have two copies R2. The enzyme has no red pigment, so it cannot able to produce the red pigment. Instead of that, the particular flower is white in color. The F1 heterozygotic condition appears R1. So the R1 is 50% only, it is functional, it produces protein. Okay, only 50%. So it need the entire percentage of protein, that is 100% to get the red color. But here you are getting the 50%. So because of that, the flower is pink in color. So the pink heterozygous contain 50% of functional protein, and that is not enough to create the red phenotypic condition, that is homozygous condition. It produces 100% of protein, so it produces red color. So this is the reason why the pink color appear in F1 generation. Next we move on to the codominance. 1:2:1. Codominance we have discussed already in ABO blood groups, that is blood group A and B both are codominant to each other. So this pattern occurs due to the simultaneous joint expression. Simultaneous means continuous expression of both the alleles in heterozygous condition. So continuously they are occurring in a heterozygous condition. The phenomena in which two alleles are both expressed in heterozygous individual is known as codominance. That is, the two alleles they are expressing in heterozygous, they are expressing AB blood group, that is AB positive or AB negative, both are codominant and they are present, they are executing the dominance. Okay. So the best examples here they had provided is red and white flowers of Camelia, inheritance of sickle cell hemoglobin, ABO blood groups system in human beings. And the codominance was demonstrated in plants with the help of electrophoresis or chromatography for protein or flavonoid substances too. So this can be seen, that is ABO blood group. By seeing our blood we can't able to find out the blood group is A or B or O or AB. So we have to test. So while testing only the process, by testing only we know the A group, B group, O group, AB group, like that. Okay, positive or negative. So like that you have to know the colors. For this you have to see the electrophoresis, which we will see in the biotechnology and plant tissue culture lesson in detail. So the electrophoresis and the chromatography, the levels where it is can be able to see the protein separation in the protein separation, how they are seen with the help of the genes. Okay. So Camelina is an example. Camelia is an example for codominance. So this one is not in your book, but I am giving it. So known as, please follow the instructions. That is explanation. So white camellia is crossed with red camellia. In F1, both the colors are seen. You can see the one side of the petal, that is, you can see one of the petal is white in color and one is in that is pink, that is red in color. So both the colors are appeared in the F1 generation. So both the parents' characters will be appear because they are dominant. Both the parent colors are dominant. They appear in the F1. So that is called codominance. Okay. So the hybrid, that is the hybrid, this one is the hybrid one, this one is the hybrid one. The hybrid offspring has both the characters of white camellia and red camellia. So this condition is called as codominance. Okay. In F1, heterozygous produces the progeny, that is with an different color, with both the colors. Okay, both the colors of flower. In F2, you get the both the phenotype and genotype as 1:2:1. So in F1, you have you get that is a different color, that is both the characters with a different colors. That is, one side it is red and one side it is white color. But in F2, both the phenotype and genotype will be 1:2:1. Okay. Lethal genes. As we talk about, the lethal means death. So if it produces death, then that genes are called as lethal alleles. So an allele which is capable of producing death to the organism is called lethal allele or lethal genes. In 1907, E. Barrier reported lethal gene in snapdragon, that is Antirrhinum species. So what happens means that Antirrhinum, that is snapdragon flower, has 3 types of genes. Okay. That is, the first is the green color with the chlorophyll. The plants compulsory has to have chlorophyll, because the chlorophyll only producing the food, that is called photosynthesis. Through photosynthesis is producing, it needs light which captures the light and it produces the food. So for this, capital C is responsible for the green color plants. And yellow green colors, that is with carotenoids, that is carotenoids means yellow pigments. Okay, example, that is how the color appearance is pale green, golden or aurea plants that has the genotype capital C and small c. White plants without chlorophyll, so it is represented by small c. So the genotype in homozygous green plant capital C and the genotype with homozygous white plant is small c. So in the next slide we are not going to talk about the capital C and small c. So we are going to see about the aurea plant. Aurea plant is the yellow color plant. So you can see the both in that is the both aurea, that is in a wait. Here the genotypes, that is capital C and small c is crossed in the first generation. You think of it like that. So it is crossed, and in F1 generation you are getting capital C, small c, that is the yellow green plant, that is aurea plant. So now you are going to self this aurea plant, that is in F1. So in F1, what are the character, that is gametes will appear capital C, small c. These are the two gametes by the two sides you are taking. And when you're putting the checkerboard, you get one green, that is one capital C, and two capital C small c, Aurea plant, yellow color plant, and one small c, and you know the capital C can able to produce chlorophyll, so it will survive. And in Aurea plant also capital C is there. So capital C is there means what is the color will be pale yellow or pale green in color. Okay, so it also produces chlorophyll, so it will survive. But in small c, it cannot able to produce that is chlorophyll. So the plant cannot able to withstand, because without food production, how the plant will survive? So the plant will die at seedling stage itself. And now we can return this. The ratio can be modified, that is 1:2:1 can be modified as 1:2 or 2:1. Okay, like this we can interpret the codominance ratio. Now, pleiotropy, a single gene affects the multiple traits. Okay, so in pleiotropy, the single genes affects the multiple traits and alter the phenotype of an organism. So, one gene is responsible for more characters. That is, for example, the protein. The protein is necessary for the growth of the hair. So, growth of the hair, color of the hair, the two genes. Okay, growth of the hair, color of the hair, and the color of the hair, you have gray color, white color, black color, like this. So many colors of the hair is there. That is also, and even the pure, that is depends on the character, even the skin color. So there are, that is one gene is controlling the many traits of an organism, that is called pleiotropy. So pleiotropy gene influences a number of characters. Simultaneously it is expressing number of characters, one gene only, but it expresses more characters and they are working as an unit. So how means, now I am saying an example. Tomorrow is an Independence Day. If the normal condition is there, normal condition is there, we will plan for a celebration. So some people will be dancing, some people will be putting a skit, that is a skit, putting a skit on Independence Day, and some people will be talk for the speech on Independence Day, like that. So, but what is our goal? There are 4 or 5 groups planning to in a different programs. But what is the thing? Independence Day, for Independence Day only you are working. So, that is a single unit. So many people are working to get to celebrate the Independence Day in a grand manner. Okay, I'm saying it is, you are coming to my point now. So in a grand manner, you are bring the Independence Day for so many programs in different groups. Like that, a single unit, that is a single gene, controls so many characters. And it works as a single unit. That is called pleiotropy. Okay. So here in flowers, that is Pisum sativum, Mendel works Pisum sativum, that is pea plant. So in that, purple color, brown seeds and dark spots on the axil of the leaves, that is purple color flower, brown seeds, dark spots on the axil of the leaves. So these are governed by one particular gene, which is crossed with white flowers, light colored seeds and no spots on the axial of the leaves. And this is governed by one single gene. And both the recessive and dominant alleles are controlled by a single gene only. So the purple flower, brown seeds and dark spots on the axial of the leaves is crossed with white flowers, light colored seeds and no spots on the axis of the leaves, is governed by that is whether it is a dominant or a recessive than the particular plant in pea plant. But it is governed by a single gene. So it is working as a single unit. The best example is sickle cell anemia. So the sickle cell anemia is for us it is a disease, but for American it is a disease, and for African people it is a dominant one. It is they adapted to the sickle cell anemia and they are living together without any problems, but the Americans have the problem of sickle cell anemia. And students, I had given some questions which are related to the topic I had discussed today. So please go through it and write the questions and ready for the ready for the test. Thank you for watching the video. If you like the video, please post a thumbs up, share and subscribe to the Science Easy Tech channel.